The name of our project is the cookie as a functional food product with essential oils from coniferous raw material for inflammation control and prevention from oncological diseases. Cookies are one of the most widely consumed food products that are high in demand worldwide. There are the basic cookie making procedures, and you can change the ingredients to create different flavors. The key thing is to soften the butter in one direction. The table shows the main bioactive ingredients and their functions in walnut cake. As we can see, the walnut cake contains a high quantity of protein, dietary fibers, macro and micro elements, and B vitamins. That's why we suggest to use it in the functional cookies to improve its food value and enrich it with bioactive substances. The essential oil will be extracted by the following way. After ultra-pure water infiltration and microwave treatment, water steam distillation is used to essential oil extraction. Since essential oil is a sensitive substance to light and oxygen, it is needed to encapsulate. So we choose high-pressure homogenization for nano-encapsulation of essential oil. The primary emulsions will be obtained by mixing 1 gram of essential oil and 1% of twin-20 in 100 milliliters of water, using an ultra-thorax homogenizer. The obtained emulsions will be subjected to high-pressure homogenization in a nano-dB electric bench top laboratory homogenizer. At the end, hot nano-emulsions should be rapidly cooled in an ice bath to crystallize oil droplets. Transmission electron microscopy could be employed to characterize nano-emulsions. The next step was chemical analysis of bioactive compounds. Gas chromatography coupled mass spectrometry was used to analyze the composition of fatty acids and essential oils. The preparation of samples and specific chromatographic conditions of GC-MS analysis procedure is described in the text. The components of oils were identified using special mass spectra database. The relative percentage content of each component was calculated by peak area normalization method. The example of chromatogram of the oil sample you can see in the upper right corner of the slide. Next, the anti-inflammatory animal experiments of essential oil was established. The experimental groups are shown in the figure, which were divided into control group, model group, essential oil group, and positive control group, while the specific operation method and those for animal experiment were as described in the text. After 7 or 30 days, the animal's tissues or sera were tested for various indicators, and the results of the subsequent experiments were further analyzed for signal pathways.
As you know that we chose the walnut as the subject of our research work. It contains polyphenol compounds which reduces oxidation and has anti-inflammation effect, improves signaling between brain cells, but also has beneficial effects against heart diseases and numerous types of cancer. In this case, we chose colorectal cancer. Factors of this kind of cancer are old age, low fiber, high fat diet, and other bad habits. In this research, 6-week-old female anthemic nude mice were injected subcutaneously with HT29 human colon cancer cells. 7 days after tumor cell injection, mice were randomized after stratification for body weight control or walnut diet. The control diet was modeled after standard AIN-76 diet for mice. It's a vitamin mix. Mice were fed the experimental diet for 25 days. On the right side on the figure you see that after injection, weight of the mice increased day by day. Down below you see the figure of food intake. Body weight and tumor size were measured 3 times weekly. 4 mm thick sections were placed on the microscope slides for histological and immunohistochemical staining analysis for further research analysis. Tumor size increased during the observation period. Final tumor weight was significantly reduced by 44%. On the right side you see that by walnut dieting, cancer cell reduced by size more than control group. In this research, miRNA of the colorectal cancer studied. miRNA may help explain the relationship between walnut consumption and decreased colorectal neoplasia risk. Several studies have documented that dietary components modify miRNA expression in pancreatic and hepatic carcinomas, implying a potential effect of dietary intake on miRNA-mediated oncogenic transformation. Walnut diet causes significant changes in the expression profile of miRNAs in localized colorectal cancer tissue, incorporates protective fatty acids in colonic tumor, providing potential protection to cancer development either through its direct effects of anti-inflammations and angiogenesis, or through additive or synergistic effects of multiple other compounds present in walnuts. Tumor size.
On the basis of chemical composition of cookies ingredients, we calculated the nutritional and biological value of cookies. As you can see, one portion of cookies can satisfy the daily food nutrient requirement for dietary fibers by 18%. The sensory assessment is one of the important steps in cookies manufacture. The color of the cookies is uniform yellow to golden shades, and the structure of the cookies is fine tissue with fine and uniform small pores broken without impurities. As for texture, it has uniform thickness, no shrinkage, no deformation, no foaming. It also tastes sweet, crisp and loose with fermentation flavor. Cookies can be nitrogen-packed with polyethylene package to prevent exposure to air. In the warehouse should be clean, ventilated and dry, moisture less than 40%, in the room temperature, and they run dust prevention, fly prevention and so on. Shelf life will be hypothesized using ASLT method based on critical water content.